Hello, everyone. I'm Dr. Carmen Puliafito, and I'm happy to have here today on retina synthesis Dr. Glenn Yu, who is professor of ophthalmology at the University of California at Davis and director of teleophthalmology there. Glenn is an authority on gene therapy of the eye, and today we're going to talk about intravitreal gene therapy for neovascular AMD. Glenn, welcome back to
Thank
Retina
you, Carmen.
Synthesis.
Thank you for the invitation.
So, can you explain to us a little bit about the basics of intravitreal gene therapy?
The concept of gene therapy overall is that rather than injecting a drug into the eye, you're injecting, I always tell patients a blueprint, right? So you're injecting a viral vector that delivers a blueprint, a copy of the genes encoding the drug, such that your cells are essentially exploited to make the drug on its own, so that theoretically, once you get a gene therapy, the eye will be making the drug indefinitely. So the intravitreal gene therapy is no different from any other gene therapy, except that the distinguishing feature is an intravitreal injection, which we're much more familiar with than a subretinal injection. The first generation of gene therapies were really subretinal, even going back to Luxturna, which is a subretinal gene therapy where you have to do a vitreoretinal surgery, you do a vitrectomy, and you do a subretinal bleb. You inject the vector underneath the retina, and then the retina will start making the drug. This, unfortunately, is very challenging for folks who are used to the ease of just coming into clinic and getting a shot in the eye. And so the advantage of an intravitreal gene therapy is that you can give it in the clinical setting, and it should be no more complicated than a routine regular a regular intravitreal injection, except that it should be a one and done treatment.
Where does the vector sequester and produce the drug?
That's a very good question. That a lot of people ask that. So, this all the gene therapies under development currently are driven by what we call a ubiquitous promoter, meaning that the gene is designed to be driven in any cell that it wants. So it's whatever the AAV vector is delivered to. So it doesn't necessarily need to get into the retina. Theoretically, you could have the gene be delivered into the iris, into the ciliary body, or into any kind of cell, not just photoreceptors, could be myeloglia, bipolar cells, ganglion cells. We just know it gets in there and it transduces cells. There are some studies that suggest that there are broad ranges of cells that are transduced, but they're limited and none of them are in humans. So we don't know exactly what cells, and that remains to be a major question in the field.
What's the time course of transduction?
So it doesn't work right away, which is one of the reasons why current gene therapy trials usually will either need an injection either immediately before or immediately after the gene therapy. We know from a lot of preclinical models that it takes about a month for the gene therapy to really reach its peak, maybe 1 or 2 months. And so if someone has previously a heavy treatment burden, you really do want to be giving them a one injection either at the time or even after the initial gene therapy to make sure they get kind of bridged until the gene therapy kicks in.
So the pioneer company in this field was Adverum, and we'd like to talk about the results of their clinical trials and the current state of their program.
Correct.
So what was their first important clinical trial?
The first ones I would say that most people know about are the OPTIC and the INFINITY trial. Those were phase 2A studies that essentially looked at patients who were previously treated with anti-VEGF injections for at least a year or so prior to the treatment. And they looked at patients who had pretty high injection burden, people who were needing 9, 10 injections per year. And they gave them a single dose of the Ixo-vec vector, which is their intravitreal gene therapy that produces something akin to aflibercept. And they found that after the first year or so, that there was a significant reduction, roughly about half of the patients can become injection-free, especially at the higher doses, and also about 80 to 90% reduction in injection burden overall. It sounds amazing, but one of the major issues that came out of those trials. So at the time, the Infinity trial was designed for patients with diabetic macular edema, and the Optic trial was designed for patients with AMD. But what they observed was that there was ocular inflammation in a few of the patients in the Infinity trial with the diabetic macular edema. And those patients actually developed not just inflammation, but hypotony that ultimately led to significant vision loss in, I think, 2 patients and early termination of the Infinity trial. Fortunately, they did not observe that similar effect in the Optic trial for AMD patients. So they've gone on to conduct the Luna trial, which is a phase 2b trial, which was a dose expansion with different types of steroid prophylaxis, including everything ranging from topical difluprednate to intravitreal Ozurdex to oral prednisone courses of various types to optimize what they think will be the dose leading to their phase 3 pivotal trial, and also the prophylactic regimen that's best for that. And I think ultimately they settled on a lower dose. I think it'll be a 6E10. So we usually say 6E, meaning 6 times 10 to the 10th power viral genomes per eye in the pivotal trial.
And what was their conclusion about dosing with steroids? What's the optimal course?
Yeah, so from their results, essentially what they found was that, first of all, the Ozurdex didn't make a huge difference compared to topical. In fact, in the Ozurdex arm, what they found was that people started to develop a little bit of inflammation around the 3 to 4 month mark, which is roughly around the time the Ozurdex would come down. So they actually changed the protocol midway to essentially bridge them with topical steroids. And so given the fact that the intravitreal and the topical seems to make not a huge amount of difference, the decision in the pivotal trial is to just go with topical difluprednate. The other learning was that oral prednisone didn't seem to add. I mean, itself as monotherapy works fine, but it wasn't that it was much more protective than topical difluprednate. And again, given the potential risk that you have to, or concerns about monitoring patients with oral steroids, they decided to just stick with difluprednate topical. And so that's the learning, was that topical difluprednate seems to work the best or adequate.
So lower dose gave less inflammation.
Generally, in all of gene therapy, lower the dose, the better. You're usually just worried about low efficacy. But one of the great things that they've done, and I haven't seen this type of results from other gene therapy programs, is that they have aqueous levels of aflibercept measured from patients out to 4 years. It's not everybody, it's just a subset of patients.
Mm-hmm.
But what I've been very, very impressed by is that if you look at the actual protein level collected from the aqueous, and this is just from the aqueous, we presume that the levels in the retina would be much higher, right? But the aqueous levels were almost rock steady across the 4 years, meaning that if your eye is making a certain amount of protein or drug on day 1, or month 1 after the treatment, it will probably be making the same level 4 years out. The big difference, though, one of the things you might have to pay attention to is that there is quite a bit of variability in individual responses. So you and I could have gotten the exact same dose of gene therapy, but my eye can be making like 100-fold less than the protein your eye is making. Because I think, unlike a drug where you inject and you're pretty much getting that level of drug, maybe some subtle variations in bioavailability. With gene therapy, it depends on how it gets into cells and what cells are making your protein. So maybe one person's eye may be making a lot more protein than the next person. But even though there's inter-individual variability, the stability over time per individual seems to be quite high, meaning that if you make a lot of drug, you'll probably be making a lot of drug for the next few years. And if I'm making less drug, I'm probably not going to change that much in the next few years too. But I do think that the idea of that could be pretty promising because it could suggest that if it's safe, we can give me, let's say if I have a lower dose or a lower production, we can potentially supplement with another, we've never really talked about, because we always want gene therapies to be a one and done, but maybe it could be a two and done. And that's not bad if it works that well. So I generally assume that's probably why a lot of these companies would rather sacrifice a little bit of efficacy for safety and long-term and having long-term durability.
So how many eyes were in the Luna trial and what were the clinical results?
Yeah, and honestly, I don't remember the exact number of patients in the Luna trial, but I can tell you that, and the results are still pending. The final results are still under analysis. But overall, the Luna trial showed that even at the lower 6E10 dose, the amount of protein that's produced, as I mentioned, many of the patients were still very high. And the outcome was overall very similar to what was observed in the optic study. So what they really showed was that they can achieve similar efficacy at slightly lower dose, but more safety. So they did not observe as much of the levels of inflammation observed certainly not in the Infinity, but not too much in the optic either. So similar efficacy with, again, roughly about a half reduction in about 80 to 90% reduction in overall injection volume and about half the patients being injection-free through year one.
Let's talk a little bit about steroids again, because this is always an issue with intravitreal gene therapy. How long will the steroids need to be administered?
So in the Luna trial, the topical steroid course was about 6 months. In other intravitreal gene therapy trials, they're roughly around the same duration. We believe that right now, all the intravitreal gene therapy trials are hovering around that same level, with the assumption that we really want to just keep that first 6 months window completely quiet. Do they really need 6 months? It's unknown at this point. I would say that it's probably highly variable between individuals because I think there are some patients that are just going to be more pro-inflammatory than others. We do know in work from, Paul Sieving's work in X-linked retinoschisis, we found that many patients with retinal diseases at baseline have already a amped up pro-inflammatory response. Like they're maybe due to violation of the blood-retinal barrier, their eyes are just a little bit more trigger happy already. And then you put gene therapy on top of it, it could potentially make things worse. So there could be some inter-individual variability. The other thing is that the way we detect or describe or report ocular inflammation as an adverse event, the more, I've been participating in gene therapy trials, and one thing I've observed is that the quote inflammation that we observe in these patients aren't necessarily what we typically see in like a real iritis or anterior uveitis or even vitritis. Oftentimes they're a little bit fine, very low grade, like 0.5 to 1 plus cell, and many of the times they're more like pigment than actual white blood cells. So we think some of this may not be the same, effect as ocular inflammation from uveitis, but more like some kind of response maybe to iris cells or pigmented ciliary body epithelial cells that are just being released by the gene therapy. And whether these type of low-grade inflammation will ultimately lead to vision loss or actual adverse effects is not well known. Certainly in the Infinity trial, those are like 3 to 4 plus real cell hypotony and eyes, losing vision. We definitely don't want that. But I think it's possible that some of these later trials, as they become more refined, they're starting to classify like pigment versus true cells. We might also see lower rates of true ocular inflammation using these kind of more modified criteria based on our new understanding of gene therapy.
How do the clinical results from intravitreal gene therapy stack up against those seen in the subretinal gene therapy approach?
Yeah, so I would say overall, if you look at the two main outcomes, most of the current trials are comparing again after a single dose of the gene therapy, what's the total reduction in injection burden and the percent of patients who are injection free. Some people also look at time to first rescue. They're actually relatively comparable. I would say that right now, if you were to say which one is better, intravitreal versus subretinal, I would say that they're relatively comparable. The differences are more not so much on efficacy, but on the practicality and safety. I think intravitreal is obviously a lot more practical, easier to do. Don't have to worry about surgery, don't worry about lens status, but there's definitely a little bit higher levels of ocular inflammation. If you look at the AbbVie trials for the subretinal ABVV RGX-314 or Surovec vector, you'll see that there's very low rates of inflammation. If anything, it will be related to just the vitrectomy surgery, a little bit of post-op inflammation. But many of those patients are not seeing kind of long-term evidence of iritis or posterior uveitis. So I think certainly subretinal injections, gene therapy is going to be safer, but harder to do. And intravitreal injections are probably a little bit easier to do, but potentially more risk for inflammation and requiring longer courses of steroid prophylaxis.
So what's the next step for Adverum?
I think that they're settled on a safe and lower dose. I was pleasantly surprised by the fact that even at the lower dose, they can achieve protein production levels comparable to the higher dose used in the earlier studies. So I think that they really just need to stay the course, really pay close attention to any evidence of inflammation and try to really tackle those and try to get those answered early. But I do think that there's some a lot of potential to this product. I think the bigger question for me for gene therapy, besides even if it ends up being safe, effective, is also going to be the practicality of incorporating it into clinical practice. I think that there's a lot of hesitancy I detect from current providers about what is this, what is the potential risk of having something you can't turn off? Like what happens if it doesn't work or it does cause a problem? There's no way you can remove the gene therapy. And I think it's a known concern. So I think the longer, the more data we have of long-term follow-up of these early gene therapy trial patients, the more confidence we'll have that that they're sustainable and safe in the long run. Because remember, one of the things about I know that there's all these tyrosine kinase inhibitors, TKIs coming out, that they're potentially be able to go all the way out to 6 months. But I think we have to really think about gene therapy in a completely different way. Every drug right now is still a drug. It needs to be, when it goes down, it goes to 0 and you have to put another one in. So even if you have a longer lasting drug, you still are constantly thinking about when or how much to give and when to supplement. Whereas I think gene therapy, even if it doesn't work perfectly, it will never go down to 0. Your eye will always be making some anti-VEGF. So I think it's more about stability and disease stability rather than durability of drug, because we're not really thinking about when the drug wears off. It's almost more about how well is the disease controlled now and whether you need more or some rescue.
So are they is Adverum in their phase 3 trial yet or not?
Currently, yes. So the Artemis trial is currently recruiting. They're are starting to recruit patients, and so hopefully we'll be seeing some exciting results in the next couple of years.
Great. Well, Glenn, this was a really wonderful review of intravitreal gene therapy for neovascular AMD and a very realistic assessment of its potential for entering our clinical armamentarium. Thanks for participating on Retina Synthesis.
Yeah, thank you, Carmen. Thanks for the invitation.